Flu shots, which you should get, contain an H1N1 strain and an H3N2 strain, as well as a couple of influenza B strains. Now, the H and the N, this notation refers to hemagglutinin and neuraminidase, and these are two proteins on the surface of the virus that it uses in order to get into cells and to get out of cells, and which, because they're on the surface of the virus, our bodies can learn to recognize and make antibodies against them. So, these are key targets for vaccination, but they're also key targets for the virus to change in order to evade our immunity. And so influenza viruses are really, really tricky, and the reason why we have to have new vaccines every year is because they can change those surface proteins in a couple of ways. They can undergo antigenic drift, where basically they just acquire a bunch of little mutations in that protein that make them harder for our antibodies to recognize, or they can undergo antigenic shift, where they actually swap, they mix and match those H's and those N's. And so each year the scientists convene and try to figure out which is probably going to be the most prominent circulating strain based on things like what was happening in the southern hemisphere. And then there are various ways that they can make vaccines, various forms of inactivated or dead versions, or attenuated or live versions. These different methods, pros and cons, I'll get into, but these basically are trying to outwit the virus, but then the virus is going to evade, and so you get this kind of tug of wars and this evolution that makes it so that we have to have these new vaccines every year, which you should get. So let's get a little more into the science of influenza vaccines, as well as kind of like a compare and a contrast to SARS-CoV-2, so the coronavirus we've all been hearing so much about. Since now we have this framework in order of thinking, we can kind of think about how influenza is similar and different.
Disclaimer that I am not a virologist and I'm not an immunologist. Speaking of immunology, there's also things like cell-mediated immunity, so things like T cells, which I'm not going to get into. I think it's easier in terms to just think about antibodies, but note that there are other ways that our bodies can recognize viruses, and the evolution can also affect the T cell recognition as well. But anyway, let's talk about how this influenza gets into cells and affects them. So as I mentioned, there's these two main surface proteins, hemagglutinin and neuraminidase. Now, hemagglutinin, that's like the spike of the coronavirus. It's what's going to allow the virus to dock onto cells. And it's going to dock onto cells by binding to sialic acid. So sialic acid, you can think of it as kind of the glue. It's this little sugar chain that gets added on to various proteins in the membranes of our cells. And this allows it, because this hemagglutinin recognizes and binds to the sialic acid, it's able to dock onto our cells. Once it's docked onto our cells, what happens is it gets endocytosed. So basically the cell swallows it, and it swallows it by pinching in a bit of the cell membrane containing the virus. This traps the virus in this little cellular compartment called an endosome. And what happens in the endosome is that the pH gets really low, so it gets really acidic. This causes the hemagglutinin protein to change shape. So it undergoes this shape shift that's actually going to merge the viral membrane with the cell membrane, this fusion step, which is going to release the viral RNA out into the cell. So we'll get more into the genetics and the composition of that viral genetic information, but it's made up of these 8 strands, at least in influenza A. It has these 8 strands of RNA, which contain the instructions for making the various proteins that the virus needs. And so these RNA gets out, the virus makes more copies of itself, takes advantage of some of the cellular machinery to do this, as well as machinery that is, the instructions are in that RNA for. It's able to make more copies of itself, make more of the different proteins it needs, and then assemble into new virus. Once the virus is assembled, it can now bud out of the cell. But remember, you still have that sialic acid on the cell surface, which is how the virus docked and got in the first place. And so if you don't do anything, the virus is just going to get stuck on its way out. This is where neuraminidase comes in. It's a sialic acid cutter. And so what it does is it's going to be able to cut that sialic acid that the virus is stuck to in order to release it. Now, You want to make sure that you don't have too much of this, or else the virus is going to cut itself off before it can get into the cell. So there's typically like a 4-to-1 ratio of HA to NA, so that the virus has a better chance of sticking and getting in without getting cut off. Neuraminidase is also important for helping overcome some of our more generic defenses. So our cells in our bodies, we have a lot of mucus. And so mucus is made up of this proteoglycan, so mucin, this protein, this mucin protein that's actually coated in a bunch of sugars, including this sialic acid. This can act as a sort of decoy and kind of stop up the virus. But if the virus has neuraminidase, it can cut itself off. Another fun fact about neuraminidase is that tamiflu is actually a mimic of sialic acid that competes for the actual sialic acid, and therefore it prevents the virus, kind of like overcomes the neuraminidase, so that the virus it has a harder time getting out of cells and getting away from those decoys. Because hemagglutinin and neuraminidase are on the surface of the cells, they make good targets for our body to be able to recognize the virus. And as I mentioned, this is also why the virus can change them. They undergo this great selective pressure where the virus has to evolve to have changes in those proteins in order to evade our immunity. So this is a really oversimplified guide to our adaptive immunity, and it's focusing just on the antibody side of things and not on the like cell-mediated T cells and that sort of thing, which are more complicated. And again, I'm not an immunologist. But what happens is that antibodies are these little proteins that your body makes. They make these proteins kind of like randomly, in that you have a bunch of these immune cells called B cells, which have the instructions for making antibodies, but they have a wide range of antibodies that they could make. And then each of those cells is going to kind of combine its DNA in a way that makes a specific antibody. So these antibodies have these unique parts, these variable regions that are going to be able to bind to different things. And so each of these antibodies made by a different cell is going to recognize a different thing. And this is just going to be randomly based on how they recombined and what proteins that they made. But the ones that actually recognize foreign things and don't recognize our own things, these get selected for, and you make a lot of them. So I mentioned the word antigenic. So antigens are just basically things that an antibody recognizes. And so each of these unique antibodies is going to recognize different antigens. And so some of these antibodies are going to happen to recognize the antigens that were on the surface of the protein, the virus, so they're going to recognize the hemagglutinin and they're going to recognize the neuraminidase. Those antibodies are then going to get selected for, so your body's going to say, oh, cool, this one's great, let's make a lot of it. And then some of those are going to fight the virus, and then some of them are going to stick around if you're actually infected then, and then some of them are going to stick around and keep watch, so that if the virus infects again, well, now your body knows it's foreign and knows to attack it. So this is the basic idea of vaccines, is to try to bypass the actual infection part and get your body to select those antibodies that recognize the virus without actually making a person sick. There are various ways that vaccines can do this, can introduce your body to those viral parts. These include conventional vaccines like killed or inactivated viruses, or weakened or attenuated viruses. So if you talk to a virologist, they'll say that you can't, or at least most virologists, I don't know any virologist that actually will let you say that viruses are live. But basically, we often talk about viruses as being live or dead. And so the live virus would be something that can actually infect you, that can replicate, so make copies of itself and that sort of thing. Whereas a killed virus would be something that's inactivated. It can't actually like infect you, or can't make copies of itself and keep infecting other cells and that sort of thing. But technically we call them inactivated and live attenuated. So attenuated, like weakened. There are also methods in that you can use just the viral proteins. You can put those viral proteins into membranes, or as we've seen a lot with the coronavirus vaccines, you can actually introduce the genetic instructions for making those proteins and get the cells to make those proteins themselves. And so this is the idea with like mRNA vaccines. And so in the future, there might be mRNA vaccines for influenza, but currently we're dealing with mainly the inactivated, live attenuated, and viral protein vaccines. These vaccines I'll get more into how these are made, but basically they contain 4 different strains typically, so they're quadrivalent. These include 2 influenza A strains and 2 influenza B strains. So you have one H1N1, one H3N2, and then a couple of these influenza B strains. These are then made into viruses or made into vaccines in these various ways. They can be the vaccines can be produced in eggs and cells, or recombinantly, where basically you put the instructions to the cells and you make and purify out the proteins. And so we'll get more into the details of how these vaccines are made and some of the pros and the cons of the various methods. But first, I just want to mention why we need to have these different strains and what these strains actually mean. So as I mentioned before, influenza A has its genetic blueprint, so it has its genome in 8 RNA segments. If you think about our genetic blueprint, so our genome, it's in DNA, and it's housed in these 23 chromosomes, these 23 pairs of chromosomes, and which are these super duper long DNA segments. Influenza kind of has like mini chromosome type of things, but they're RNA. So influenza A, it has these 8 strands, these 8 RNA segments. And there are these each encoded, instead of having like tons and tons and tons of proteins encoded in each strand, so have the instructions for making lots and lots of proteins in each of our chromosomes, they only have the instructions for making a couple of proteins in each of their strands. And so this strand 4 has the instructions for making hemagglutinin, and the strand 6 has the instructions for making neuraminidase. And then there can be different versions of each of these genes. And which version of these a strain has is going to determine the subtype. So this is how we get things like H1N1 and H3N2. So those are currently the most prominent subtypes that are circulating among humans, but there are actually 18 HA subtypes and 11 NA subtypes. Thankfully, not all of them infect humans, at least not yet, but they have the potential to, if they were to evolve. And so this is one of the reasons why it's important to have surveillance of animal influenza and that sort of thing in case those viruses evolve in a way that then they can infect humans. These strains are negative sense. So this means that basically in order for our bodies to make proteins from them, you need to make another copy of them. So in our bodies, we have, typically what happens is we have a gene and it's in DNA, and then you make a messenger RNA copy of that, and then you make a protein from it. In their case, they have an RNA recipe and they have to make an RNA copy of that RNA recipe, and then they can make protein from it. So this is basic. They do this they make the RNA copy that has the instructions for the proteins using the RNA-dependent RNA polymerase, so this RDRP. It's going to make the positive sense strand, and then they can make the ribosomes. So the cells, proteins will make the cells, machinery will make protein from it. And so this is an extra step if you compare it to the coronavirus, which is a positive strand virus. It doesn't have to undergo this step in order to make proteins, but it still does need an RDRP in order to replicate. But the influenza virus, basically, it has to go through this step of making an RNA copy before a protein can get made. This is a little technical detail, which also influences, this RDRP is also going to influence mutation rates. Speaking of mutation, so mutation is one of the ways that the virus can evolve to get around our immune systems. And basically what happens is that every time the virus replicates, so makes a copy of itself, the RDRP, that RNA-dependent RNA polymerase, the thing that's making copies of its genetic information, it can make boo-boos. So it's not perfect, it's going really fast. And in the case of influenza, it doesn't have proofreading abilities. And this means that it can acquire mutations over time. With antigenic drift, you have these mutations in an antigen's gene that build up over time. And what happens is that those antibodies become gradually less and less effective. And then ultimately they don't recognize that original antigen, but it happens gradually over time. But it does happen a lot in influenza, especially because the RDRP, the influenza's RNA copier, doesn't have proofreading ability. And so it's going to acquire those random mutations. if those mutations offer a benefit to the virus, such as maybe they make it so that the antibodies don't recognize it as well, or maybe they have another benefit to the virus that just happens to affect the antibody binding. So maybe they make it so that the virus can infect cells more easily, or make more copies of itself. Then those viruses are going to have a growth advantage. They're going to outcompete the other viruses and therefore be selected for. Whereas if a mutation actually harms the virus, makes it harder for it to copy, makes it so that the antibodies bind better, this sort of thing, it's going to have a fitness disadvantage. It's going to be selected against, and it's going to die out. And so in this way, you're going to get this drift of these antigens, of these surface proteins that the antibodies would normally recognize, into a form that our bodies can't recognize anymore. So this is antigenic drift, and it happens slowly over time, but not so slowly, especially for influenza, because of that highly error-prone polymerase. There's also a more sudden change, which is called antigenic shift. Now, this happens when different influenza viruses swap genetic segments with each other, and this leads to a sudden and dramatic change in antigenicity, so that all of a sudden our antibodies can't recognize those antigens anymore. Before, it was kind of like they just gradually got less and less familiar to the point where we couldn't bind them. So you might get weakened binding and then eventually no binding. But here it's just like all of a sudden no binding. What's happening here is you're actually swapping out strands. So you're mixing and matching those H's and N's from different influenza viruses. This can happen if a cell gets co-infected with a couple of different viruses, and then the gene segments get reassorted. So remember, you have those different genetic segments, which makes it kind of easy for the viruses to mix and match. And the resulting virus is going to have a new antigenicity. This is really dangerous because it can lead to pandemics or animal influenza spilling over into humans. So maybe not all of these are compatible with humans, but you have a virus that's infecting, say, birds, and then some of those strands mixed with a human virus, you can end up with a virus that is more easily able to infect human cells. And so there are ways in which these new genes can get introduced into viruses that are better at infecting humans. And this can lead to like spillover and pandemics and things. So you can also have, it doesn't have to be that you have different subtype numbers. So the different subtypes can be different enough that you can get antigenic shift even within a single lineage or subtype or whatever you want to call it. So basically you can have multiple H1N1s combine with one another and then make a new H1N1, which is still called an H1N1, but it is antigenically very different. And this is one of the things that happened with the H1N1 pandemic, where you had these recombination that led to different antigens being displayed, then the population had immunity too. So how do we get that immunity? The main influenza vaccines are inactivated, so those like dead virus or killed viruses, and live attenuated viruses. So those weakened viruses that can infect, but they can't really make you sick. Most of them are inactivated, but the live attenuated includes like the flu mist, so that nasal spray. I'll get more into the different vaccines that are used, but here's an overview of how these are made. So you can have the inactivated virus. So basically this killed virus. These viruses can be killed with various methods. And then they show your body all of the viral proteins, but your body doesn't, they don't have the capability to replicate and cause disease and that sort of thing. So your body's able to see it, but not get sick. And then the attenuated virus vaccine, so this weekend, this like live virus, although remember we can't really say virus is live, but basically these can infect cells, but they can't really cause disease unless you're immunocompromised. In either of these cases, you're going to have to grow up large amounts of the virus and then either weaken it or kill it altogether. You can grow up the virus in either eggs, so typically chicken eggs, or in mammalian cell culture. What happens is the way that they do this is that they take a virus that's been adapted to grow in the cell culture. So growing in cell culture is not quite like growing in an actual person. And so the virus is going to adapt to be grown in those different cells. Now you can take a virus that's adapted to grow well in those cells, and now you infect it with the strains of the virus that you want to you want the vaccine to include, that you want the virus to include. So you take those viruses that you want and you infect the cells or you infect the eggs. What's going to happen is you're going to get that genetic reassortment. So the strands are going to swap in. And what's going to happen is that then you have to select the ones that you want. So you select the ones that have the H and the N that you want, but they're going to have the background of that adapted, that culture or that egg adapted virus, which is going to make it so that you can make a lot of it. If you just use the wild type, it would be harder to make as much as you need. So by using the adapted virus, you're able to get the antigens you want, but in the background of a virus that can produce more virus more efficiently and that sort of thing. You can then purify out the ones that have what you want and infect more eggs or infect more cells and let them make more virus. And then you can collect and then weaken or kill the virus and administer it in the case of the egg-based vaccines, or in the case of the cell-based vaccines, you actually purify out those proteins or purify out the antigens and administer them. Some pros and cons of the cell versus the egg-based. Basically, traditionally the egg-based are used. It's cheaper, conventionally used. Some people are allergic to eggs, and so then they might want to use a cell-based vaccine. Another reason why sometimes cell-based vaccines have a potential benefit is that with the egg-based vaccines, you're growing the virus in the presence of the chicken selective pressures. And so these are going to have things like the chicken sialic acid. So the virus might be adapting to be recognizing the chicken egg rather than recognizing the human. And so if it changes its antigens, if it changes the HA and the NA in a way that makes them recognize better, like bind better to the chicken sialic acid than say the human sialic acid, well, now when we put that virus or we put that into humans, it's going to be more like, it's going to be different from the form of the natural form. And it might be different enough that our bodies are making antibodies against the wrong thing. So this is a potential downside of using these egg-based techniques. Even with cell-based techniques, you can also get changes to being adaptations to the cell culture, even though you're having them put in the antigens, kind of like those main antigens you want after the cells are back already adapted. So it should just be the background that's going to be adapted to the cell culture. You're still then having to grow up this combined virus, and it has a chance to be adapting to the cell culture, which can be changing the antigens. And so then when you go and you purify out those antigens, they might be slightly different. One way that you can make it so that those antigens are going to be what you think they are, and don't give them a chance to adapt, is by doing recombinant expression. So basically recombinant is where recombinant technologies where we stick the genetic instructions for making something into cells and get those cells to make it for us. And so we're recombining DNA. So we recombine the DNA, the instructions for making the hemagglutinin protein, say, we stick it into a circular piece of DNA called a plasmid, and we can stick this into expression cells, such as insect cells, get them to make a lot of that protein, that HA protein, purify them out, and inject them. And so this is the benefit of a genetically defined antigen, so it can't undergo any genetic changes. But a downside is that it's a lot of work to make and purify all of these, and then they could have different like glycosylation and stuff. So the insect cells might modify these proteins differently, even though the underlying protein sequence is the same. And so each of these vaccines is going to include virus or at least antigens from 3 to 4 strains. And so typically this is 2 of the influenza A strains. This year, 1 H1N1 and 1 H3N2, as well as 2 of the influenza B strains. So these are the influenza vaccines in use in the US in 2022 and 2023 season. So you can see that there's a number of these standard dose egg-based vaccines. There's also a high dose egg vaccine. There's Fluzone high dose quadrivalent, often given, recommended for people over 65 years. There's also cell culture based vaccines. So there's Flucelvax. There's also this egg-based vaccine that has an adjuvant. So basically it has something that's going to make it more immunogenic, to make it so that the body is going to mount a stronger immune response to it. So basically it's the same underlying virus, but then it has this little adjuvant, it has this extra chemical that's going to wake up the immune system and say, hey, I'm putting something in you. You need to respond to this. So this MF59 adjuvant, basically it's this like squalene water. So basically it's just like this oil that makes it so that the body is going to respond more strongly. And so that again that's one of the options for the older individuals. You also have this flu block, which is a recombinant HA vaccine, so we're actually making and purifying out those proteins. And then you have this live attenuated egg-based vaccine, this FluMist, which is a nasal spray. That's the basics of the influenza and influenza vaccines. I just want to do a quick comparison to the coronavirus, so to SARS-CoV-2 and some of that. So basically the coronavirus, when we talk about a coronavirus, it's actually this whole family of viruses, but basically SARS-CoV-2 is the one that everyone's referring to when they say coronavirus these days. It, like all coronaviruses, is an enveloped single-stranded RNA virus, and influenza A is also an enveloped single-stranded RNA virus. But the coronavirus, it has the positive sense RNA, so it doesn't have to make a copy of its RNA before it actually makes proteins. This is what I was talking about before, kind of not explaining very well, but the coronavirus, it can get straight to work making proteins, or getting our cells to make proteins for it. It has its genome in one place as opposed to 8 places. So this makes it less susceptible to antigenic shift. So it can't like swap out whole pieces of it. It can't swap out like segments of its genetic information. So with influenza A, because it had its HA and its NA on these different segments, it can mix and match these HA and this NA when multiple viruses infect the cell. Now, with coronavirus, it can still undergo like recombination, where basically regions of, if a cell is infected by multiple viruses, regions of those can swap, but it's not as easy as, say, with the influenza, where you can swap whole segments simply by like whole, like actual like pieces, separate pieces. Another reason why influenza is able to evade really well is that, remember, it's RP, so it's RNA-dependent RNA polymerase. So the thing it uses to copy its genome at a high mutation rate. It doesn't have proofreading ability. The coronavirus's RNA polymerase has proofreading ability, so it has a lower mutation rate, but it still does mutate. And especially when you have so many people being infected around the world, these mutations can build up, especially if you have someone that's, say, immunocompromised. You have these long cultures, there's this high selective pressure, and you can get mutations building up. So mutations can happen, but in influenza A, they happen a lot. They happen at a very, very high rate. The major surface antigen of the coronavirus is the spike protein. And the major surface antigens of influenza A, we have 2. We have hemagglutinin and neuraminidase. We have this one main target for antibodies of the coronavirus, at least for neutralizing antibodies. So there's antibodies that are actually going to bind to the virus before it can and prevent it from docking onto cells. And so for influenza, we have this hemagglutinin, which is what does the docking, and the neuraminidase, which is what does the undocking. So if your antibodies are able to recognize those, then the virus isn't able to either get into cells or to get out of cells, unless it can't infect other cells. So it neutralizes the threat. So the docking with the coronavirus happens when this S protein, the spike protein, binds to the ACE2 receptor. And with influenza A, it's when the hemagglutinin binds to the sialic acid receptor. In both of these cases, you need a way to actually fuse with the membrane. So we talked about how with influenza, it kind of gets endocytosed. So it gets trapped in this intracellular bubble. This intracellular bubble, the pH is going to drop, it's going to get really acidic. This causes a shape change in that HA protein that causes it to fuse with this endosomal membrane and allow the virus to release. In the case of the coronavirus, in order to undergo that shape shift, the spike protein actually gets cut. So it gets cut by a protease. This can happen at the surface of the cell with like TMPRSS2, or it can happen in an endosome by a protein called cathepsin. And so here it's the cathepsin that's actually causing the shape shift by cutting the protein or the TMPRSS2 in the case about the surface rather than a pH change that's causing it. But in either case, you're getting this fusion and the release of the viral content. And so, yeah, so basically that is I find it's kind of helpful to do a little comparison since we're all used to thinking these days in terms of the coronavirus, how the coronavirus and the influenza viruses are similar and also how they're different. And the key things to remember about the influenza virus are that it has those two main antigens, so those two main things that our body makes antibodies against, the hemagglutinin and the neuraminidase. And there are different versions of these that can mix and match and also evolve in order to make it so that our bodies can't recognize it as well. If these mutations happen slowly over time, we call it antigenic drift. And if these mutations happen suddenly, such as with the swapping of the H and the N strands from different viruses, we call it antigenic shift. And both of these make it really hard to predict what viruses are going to be circulating, as well as, well, then those viruses can change when you circulate. So we end up having to have flu vaccines that are updated every year in order to help protect us from the flu. And even then, they're not perfect, but they're better than nothing. And even if they don't prevent you from getting the flu, they still can have a dramatic impact on making it so that the outcome is less severe. So go get your flu shots and hope you stay healthy.